So the plan for today is that either we go too far or we don't go that far. So we'll see how it works. So either we finish very early or we don't get to the end of it. So we ended yesterday with this notion basically of how to distinguish adaptive landscapes based on the statistics of phenotypes over time, right? So the idea here is you go sample phenotype statistics from populations at different time points, tau being the time lag between, so evolutionary distance between populations. You go measure the divergence between phenotype averages, so that's the average divergence, and divide that by the mean diversity of each population and multiply it by some sort of genomic amount of diversity, and build this ratio, plot the ratio as a function of time. Depending what type of curve you get, you're either neutral, so if it saturates to 1, or in their stabilizing selection, so the lower you are, the more constraint you have locally on your trait. Or you can be on adaptive evolution, meaning that at sort of larger time after this inflection point, you start this divergence diversity ratio starts growing linearly with time. And one thing we get out of this is we can characterize the statistics of fitness C-scape in this case. So C was the curvature, and V was basically the diffusion of the peak of the fitness C-scape. So this was sort of the statistics we cooked up, but the whole thing we wrote down yesterday, right, was basically a probabilistic description of joint dynamics of population and the fitness peak. So this is just a summary statistics of that dynamics, but in principle we have, if you say this is gamma at time t plus tau, E star at time t plus tau, condition on gamma at time t and E star t. So we know how this propagator looks like. We didn't write it down yesterday, but you can guess that it should depend on these statistics, and it's a Gaussian propagator. And because we don't have a feedback, basically E star dynamics drives the evolution of gamma. So this ends up being, you can sort of simplify it a bit further. Right. So basically, the only thing that matters here is your gamma at time t plus tau is informed by what your E star was at previous time point, how much your E star changes, so basically, which is captured by E star at t plus tau, and also by gamma at the previous time point. So you can characterize these propagators. And the first one is basically the Gaussian propagator for your fitness fluctuation, and it's independent of gamma. All right. Then we introduce the fitness flux by looking at the amount of upward sort of moves in a population in principle, in general, in a time-dependent fitness landscape, right? In this case, what we emphasized was that the total amount of adaptation is not equal to the amount of fitness gain, so not equal to the endpoint minus the beginning point, because it's out of equilibrium process, and it depends on the history. And then what we had was that in this model, we had a fitness flux that we can characterize based on the shape of this curve. It's the sort of green piece divided by the blue piece, and it's also equal to the curvature times so this is a cumulative fitness flux over time in a stationary state, so it should be proportional to this time window you look at. So the more you adapt, the more you accumulate fitness, accumulate adaptation, right? So it's proportional to the curvature, so that defines how much constraint you have and how tightly you can follow your fitness. It depends on the fluctuation, so how much you move actually, and the time, because it's a cumulative value. All right. The other thing we argued is that these quantities are built to be dimensionless, especially fitness flux. So if I give you a number for gene expression that we'll look at in a moment, or for height, or height is not necessarily a molecular phenotype, but binding affinity of proteins, things like that, you can compare these. If I give you fitness flux across different species, you can still compare them with each other, although the units of time might change, and you need to actually characterize these things well. All right. So the plan for today is to take whatever we learned so far and see what we can get out of data. All right. So we start with gene expression in Drosophila. And you probably by now know what a gene expression is, but why do we care about evolution of gene expression? So just in a nutshell, how the whole thing works, you have this promoter, transcription factor interacting with promoters, enhancers, all these sort of units that Leonid talked about, and they somehow signal to RNA polymerase to transcribe a gene, right? So the yellow thing is a gene. This is an RNA polymerase, and depending on what's going on here, you signal RNA polymerase, you produce a RNA or mRNA, you then translate it to amino acids. This translated amino acid will sort of eventually, hopefully, fold into a protein. And the protein is a sort of unit that actually does the function. So it determines what features your organism has. So, and then the amount you produce the protein also determines, for example, how much pigmentation you have in a specific organism, and that for pigmentation in particular is very important for mating, and so that directly determines, for example, the evolutionary fitness of the organism. So it still is a bit detached from what a real fitness is, so the growth rate of the organism. But for now, what we're trying to, but it is still better than looking at individual binding sites, right? So a slightly higher level trait. It's not a full fitness, so it doesn't capture everything that goes on in a cell and in many cells together, but it gives you some proxy of how things work. And so depending on reproductive success, you get basically certain types of genome being amplified in a population. They propagate, and so this sort of story goes on and on. All right. So the data we're going to look at in a minute is the sort of evolution of 7 Drosophila species. They cover evolutionary range of 40 million years. You remember the first slide I showed you was about 4 billion years, so this is much shorter. 40 million years. The feature that this data has, so let me walk you through what this tree means. Does any of you know how to read the phylogenetic tree? All right, very few. Okay, so the idea is the things that you sample in the world are the endpoints, right? So these are the sort of species that exist. And but what you try to do is to try to reconstruct a past based on commonality, for example, in their DNA sequences, right? So if you have a finite time if 2 species are very close to each other, there hasn't been enough time for divergence of sequences between them. And so sequence divergence acts as some sort of molecular clock. It has some subtlety there, so depending where on the sequence you look at, clock moves faster or slower. So you want to look at places that ideally are neutral, because you don't want to bias your clock based on how much selection is going on. So if you have a strong selection for changing a region in the DNA, you see a lot of changes. You could argue that there's a lot of there's a large time between these two, but it's actually not, right? Or the other way around. So if you have a conservation in a region, so if the region is very important, you think that the divergence has been for very short time. So you kind of need to be careful about this. Yes?
So this supposes that there is an intermediate region while speciation happens where one breaks up into two. But is it also possible that the period of speciation is actually shared between three eventual species, that the coalescence time is actually at that point?
It's possible, and it's probably the case in most of species. In this case, these guys are 7 sampled out of 40 million years, and there are a lot of little fly species in the middle. Okay, so these guys are flies, fruit flies, right? I did not mention that. So this Drosophila is a fruit fly. So there are a lot of little species that their names are not here. So these are the guys that we know the genetics of that. Alex mentioned what that means, right? So the things we study in the lab, and so they're very well characterized. But you can easily argue that probably there has been a lot of sort of splitting here. Many of them haven't survived, so we don't observe it after, a couple of million years. And the speciation, it sort of requires these species go far away normally and have enough distance that don't care about each other, and so they diverge, right? So the trees we build in some ways are somewhat maximum parsimony with some sort of, corrections to that. So the idea is it's unlikely to find triplet splitting that we're confident about in these types of trees. If you look at trees of viruses, for example, like influenza virus, that evolves much faster, and we have much denser sampling of the influenza. We sort of sequence influenza from hospitals every year, so WHO does that, and you collect this sequence and you try to build these trees. You have far denser sampling, and then you need to sort of reconstruct millions of trees, right? And so some of them for sure have triplet or quadruple 4 pair splitting and things like that, right? So this is not a worry here.
So can you, since you're talking about trees, there's different trees, right? There's the trees where
It's not me. Okay.
the terminal branch points are aligned, and then there's ones when they're not aligned, and
Right.
there's the ones where it represents the number of mutations
Yep.
and time. So can you?
Yes. So it's going there. All right. So normally what we do here is so the y-axis is normally meaningless. It's just ordering of these species. But the x-axis, you can put different things on it. You can put real time. You can put number of mutations. And real time is a bit difficult when you look at species trees. But if you look at influenza tree, for example, because we know when we sample thing, it's a slightly better defined thing. It doesn't mean when you sample it, that's the time that the strain has showed up. But you could try to sort of model this process a bit and correct for that. The other thing that is happening, it's a 40 million years of evolution. So that means there has been a lot of things going on in this species apart from speciation. And, some species, for example, Melanogaster here, which is a fruit fly in Africa somewhere, has gone through bottlenecks. And we know that from other population genetics studies. So that means the population size of the Melanogaster has gone very small at some point and then started growing. And what that means for inference of trees is that, we said the population size or effective population size was a measure of time. That's what we argued in second or first day, right? So it was a coalescence time in the asexual regime, but you can sort of build up a similar measure here. So if your population has gone through sort of bottlenecks or demographic changes, then it's as if you're changing basically the time scale of that branch. So now if you want to put all these species on the same tree and do other comparisons between them that are on the same grounding, you need to sort of squeeze the time for Melanogaster, for example, a little bit to account for these bottleneck effects. Yes?
Just to clarify for myself, is the effective population
honest, I don't want to say yes, but I can get back to you on that. I think it might have stabilized by now, but I'm not 100% sure, so I don't know. Right. Do you know? No. All right. So this is sort of a tree that's not built in a kindergarten by just kids drawing lines. There's a huge consortium of people who work on fruit flies, and there's a lot of work done on this, looking at characterizing these bottleneck events is not easy, right? So there's quite a bit of sort of statistics and modeling going on to characterize these events based on different events you see different parts of the genome. And so that's how you produce these trees, and you try to sort of rescale these branches. That's why they don't end in the same place, to account for these different statistical features. Yes?
Do you have an idea of like how many mutations would, that would make, I don't know, like a pair of these species be
so that's the scale, right? So so between Melanogaster simulans you have less than 10% divergence, right, of the genome. And this is sort of in the, so after you do the correction.
What does that mean? Because it doesn't mean that this 0.1 mutation per this length, right?
Sorry, it's not precise, but all right, so if you look within a species, diversity is 1%, right? Mu n is 1%, so within population of sort of Melanogaster and simulans, so that's the data we have, is 1%. Simulans and Melanogaster has diverged slightly more than polymorphism level, so it's about 4 million years, or 8 million years. And that's less than 10%, around 5% divergence of the genome.
So that means that in 10% of the genome, they're different.
Like 5%, yes. Yeah.
that was the pair up there, right?
Yeah. So how you read this is you sum up these guys, right? So you have a common ancestor here. I think it's like 8% or something. So you have a common ancestor, and then everything before the common ancestor is common between the two species, right? And then everything after is a split, right? So things that happen here don't happen here, because otherwise you put them together in the ancestor. Yeah.
We're saying divergence at 10% level, that's the whole genome, not just coding regions?
Yeah, so it's a neutral region of the genome.
Neutral.
Right, so and that doesn't mean all the non-coding regions. So normally what you actually do, because you don't know what's going on in the non-coding region, there are promoters and there are a lot of other things, we have no clue what they are. And it's hard actually to align them because there's a lot of movement going on. So what you do practically, you align genes, homologous genes in these species, then you look at synonymous sites, but not all synonymous sites, because there are synonymous sites that if they change, so you know what the synonymous, non-synonymous is. We have a codon that is 3 base per long. Not all the changes in that codon change the amino acid, right? And so the ones that don't change the amino acid are called synonymous. So the product of that event is synonym to the other one. But then there's things that called codon usage bias, and so they're sort of dependent on where that event happens. So some of the sites are more neutral than the other ones. And so they're called fourfold synonymous sites for some reason. I think there's a good reason for that, but I don't remember why they're called fourfold. And that's a very neutral region. So that's how you do the counting. Right. I answered your question, right? Or was there something more to it? All right. So the interesting thing about this tree is if you compare pairs of species on the tree, right, you can sort of cover a broad range of time divergence between them. So Melanogaster and simulans are pretty close to each other, but then you compare Melanogaster and Mojavensis, for example, this is a much larger time scale. And if you remember, we built up these curves. So this was our neutral curve, and there was a saturation time for neutral curve, which was equal to 1 in units of 1 over mutation rate. So if you want to characterize something meaningful about sort of divergence curves, you need to cover a broad range of times, things that are smaller than tau equal 1 and things that are larger than tau equal 1. If you now turn on selection, this saturation time becomes shorter, right? Maybe somewhere here. And then if you have adaptation, it just starts growing up here, right? So imagine you only cover a time around this area. You basically have no access to what's going on because all these curves more or less look the same. So this is sort of the thing that, characterizing the shape of the curve and really looking at how this divergence diversity ratio relates to the divergence time in ways that are meaningful for population genetics is basically the work that we did. And previously what people used to do was just comparing the endpoint of these curves with each other. And you can see that it's kind of meaningless if you just do the divergence diversity ratio at some arbitrary time here, right? And then let's try to compare these two curves, or let's try to compare a curve that has a much stronger selection and also similar adaptation. So you have no idea how to compare that. So that's a way sort of to add model, and why modeling in some ways are good for understanding anything about these processes, right? All right, so we need a range of time scales to sort of cover this axis, and this is a phylogeny that provides that. You can argue there are many species that can do that. The problem is that we don't know the genetics of many of them, and so Drosophila is actually a unique set of species that we know the genetics in a lot of these species, and also for some reason they cover a broad range of times. Okay, so the data we got here that I'm going to show you is about 6,000 genes, homologous genes in each across all these species. So 6,000 times 7 times the number of individuals that we sample from each species. That's what we have right now. So this is a sort of work that we did some years ago. Right now we did much deeper experiments and we have much more data on this, but that's for another time. All right. So let's try to build a curve. So there are error bars on this curve, and they're very small, so that's why you don't see it. But the idea here is there are sort of ensemble average values of divergence and diversity. And ensemble average means you have to run evolution multiple times to average over the evolutionary process. And unfortunately, that just hasn't happened yet, or we don't have access to the samples. But what we can do is we can sort of think of the zeroth order approximation, think of genes as different realizations of evolutionary processes. It's not a correct assumption, but it's a zeroth order assumption. So that assumes genes are independent, and there's some sort of common fitness landscape or seascape that governs their evolution. You can then split genes based on sort of functionality of them, and then look within each functional group how things vary, and that's also a valid thing. The only thing is that we need enough number of sort of samples or realizations to compute these statistics. And enough means about a couple of hundred. Yes?
Okay, so these divergence times, are they measured with respect to the common ancestor?
So yeah, I'll go there in a minute. The other thing that we had on a tree I should have had a slide on this, but I don't, is that, all right, so we don't have also a time series of the species, right? So we only have the endpoints, and we can reconstruct the tree by other means based on sequences, not based on gene expression values, based on only sequences. And the divergence times, in a sense, is basically, okay, if you look at Simulans and Melanogaster, they have a divergence time that is sum of these two branches, right? And then if you look at Yakuba and Melanogaster, in isolation, the divergence time is this branch plus this branch plus this little branch here. But then things are not independent. So if you characterize Yakuba Melanogaster divergence and then Yakuba Simulans divergence, these two data points are not independent because there is a correlation between Melanogaster and Simulans. So you kind of need to be careful as how you're grouping species with each other. And for that, you basically need to characterize these clades, and the clades are these sort of groups of species under these triangles. So these two give you one time point, and it's okay to calculate it in isolation. Yakuba, on the other hand, needs to be sort of estimated, the divergence of Yakuba in gene expression level and in time needs to be estimated by averaging over divergence of Melanogaster and Simulans. So you can sort of characterize the clade time. Let's say we have a clade which is these three species. Let's call it 0, 1, 2. Well, how do I say that? So, okay, this guy is 0. That's easier. Tau 1, Tau 2, Tau 3, Tau 4. You can build up a good notation for this, but let's for now just have a single example. So if you want to characterize the divergence time of this clade, what you do is you should evaluate it as Tau 1 plus Tau 2 plus 1/2 Tau 3 plus Tau 4, right? And then if you want to characterize the divergence of expression level between sort of, you can do pairwise divergence between 0, 1, and 2, but that's not right. What you need to actually do for this clade, this would be the divergence between 0, 1 plus the divergence between 0, 2, 1/2. Right, so you calculate some sort of averaging between things that are on the other side of the clade. Yes?
Would you weight them?
No, so the weighting comes from the time, right? So the x-axis is weighted by how much time they have, and then the y-axis is just the gene expression divergence. So you should be able to evaluate it without knowing the times. Otherwise, you're biasing your y. And so you can do it for each split, right? So this guy, it needs to be its divergence needs to be calculated against the average of the other side. And so you do it for every clade. Now you have a splitting in this clade, so these two themselves form a clade, and then you calculate the divergence of this clade against the other clades, right? That's the total divergence. So in that sense, you can build values on the y-axis that are independent of each other and values on the x-axis that are independent of each other, so you don't have biases because due to correlation on your tree. Is this clear? Good. All right, so these are the names of these clades. So Mel-Sim is basically this red clade, Melanogaster simulans, and then Mel-Yak is the larger sort of purple clade, Yakuba to Melanogaster, and so on and so forth. And so the sort of the last clade is basically the black one at the end. So this is the Drosophila genus clade. And so the divergence time, as I said, is measured in units of 1/mu, and there are numbers here. And so you can see that we have small time scale values, and also we have values that go beyond mu, 1/mu equal to 1. right? So we are able to sort of say things beyond neutrality, because that's when the saturation, the curve saturates in neutrality. And just to sort of build up the curve, we can sort of characterize this divergence as a function of time, and we can see that the saturation that we fit to this curve would basically be slightly before the melanogaster simulans, or of the similar time scale. So this is sort of a lucky situation. You could say you could have a curve that bends here, but then that would suggest that you have a larger driving rate in a system. And so this would give you the most conservative measure of adaptation, because the later you bend, the smaller the angle would be on the green curve. Yes?
Okay, so what is being fitted here? I mean, so you calculate the divergence diversity ratio. That is calculated from the tree that you have before.
This is from the data, right? So an average over all this. And then what you fit is the sort of theory we built up last time. So we know for different values of C and V, right? So these are the parameters that you fit, what this divergence diversity ratio should be. The number we have is a mu, so omega of tau was 2 times mu n. This we know from the genome. It's about 1%. This we evaluate from, the dots are from the data, and we know how the theory, what the theory tells us this ratio should be for different values of C and V. So this is a function of C and V, basically. Right, so and these are the things that are fitted. So it's a 2-parameter fit. Yeah.
And they're not degenerate. They're only like, is there?
Yeah, they're in the. Yeah, no, they're not degenerate. So basically the saturation, right? So the time scale of saturation gives you 1 over C. That's what we talked about last time. And the angle here gives you V, right? So in no way, so that's basically, if you want to eyeball it, that's how you read off these numbers. And so the green area at the end of this, right, so is basically the total, the V times tau, the total sort of number of adaptive walks that you have, and the end point here is gives you 1 over C, right? And what we had last time, and not here anymore, is the fitness flux is the ratio of the two, right? And the number is not. Right, so it's basically you have a significantly large fitness flux of around 2 if you divide 63% by 37% here. Yes?
Just kind of as an interpretation, so if you didn't have the 0, 0 point, right, and you just extrapolate the straight line,
Yep.
it would cross the y-axis at some point. So what's the interpretation of this? So like two species that basically have no time to diverge, basically with the same species, but in slightly different environments. Is that kind of?
So let me elaborate on that. So 0, 0 time, what is it? It's basically samples that come from the same species, as you said.
That ended up sort of kind of crossing basically your 1 over C.
Right. So yeah. I'll get to it in a minute. Okay. So if you go and within a species sample individuals there, right, and evaluate some divergence between isogenic individuals, right, they're still non-zero. It's a gene expression. You get a non-zero value. The diversity you get is also non-zero, right? So that's the delta we have. So you kind of need to correct your curves for that, right? And set it such that by definition your 0, 0 is equal to 0. And so that we have done, so it's shifted. So
So that you can draw individuals from two populations, create kind of shuffling them.
yeah.
Create the subpopulation.
I mean, we have polymorphism in this data. So we have within population measurements, and we have isogenic measurements, meaning same genome but different times measuring the gene expression values, right? So polymorphism means samples from a population that are not necessarily having the same genomes, right? So they differ by mu n, L being the length of the genome. And isogenic means they're identical genetically as much as you can make them identical in the lab by doing inbreeding. And so you have to do many rounds of inbreeding to actually make them identical. And you can argue inbred species may not be the healthiest ones, but well, that's what you can do for flies. But yeah, so this is already corrected for all those effects. Okay, so this was so far averaging over many genes, assuming each gene is one realization of an evolutionary process. Yes?
The divergence values are measured across the entire genome, not just the coding regions, right?
These are gene expression divergence, right? So you just count how many mRNAs you got.
Okay.
It's numbers, right? So there's no genetics here at that level. The only time genome comes in is mu n in front. All right. Okay, so one thing we have here is we have this time scale separation, right? So we have evidence for macroevolutionary adaptation that sort of populations have a strong enough stabilizing selection to stabilize within their own environments, and then there's adaptive drive by environmental fluctuation or whatever it is that drives them on much longer time scales, and so that's what you see along the evolutionary. So that's the evidence. It's coming from averaging over many genes, and so you can guess that genes have different modes of evolution. And because we have basically a probabilistic description of these processes, we have a way to go and ask for individual gene in a probabilistic way, what's the characteristics of its fitness seascape, right? So this is sort of average one. And the idea is I can build up a propagator of observing a value of phenotype or mean phenotype given what I know what the ancestor of phenotype is and the divergence time between them and sort of the parameters of the model C and V, right? So this is the thing we can evaluate from the model. So this is what the theory gives us, and it is marginalizing over all possible scenarios of the environment. Okay. So this would be one line of evolution. In real sort of stories, what we have is these sort of trees, right? Things don't go over a line, rather they're splitting, and you need to account for the topology of evolution. So you need to compute a propagator along the whole tree. Yes?
Could you remind me what the subscript
Ancestor. So what you would do is, so the ancestor would be basically any split point, right? So you start, so what's the sort of probability of seeing a specific gene expression value here, given that my ancestor was here, right? And we don't know, it's not a line. We need to account for the tree, so you basically need to do the propagator and multiply along each sort of individual branch and then do a branching. At that point, define a new ancestor. So it'll be the number of lines on this tree give you the number of sort of multiplications you have to do for your propagators. The other thing we don't know is we don't know what the environment was, and that's why we are doing the integral here. We also don't know what the ancestors were, right, at each split point. So you need to basically integrate over all the unknowns to characterize what's the probability of observing the endpoint gene expression values given all the parameters, so given basically the specific C and V, the curvature and the fluctuation, while integrating out all the unknowns on the tree. And since this thing is Gaussian, you can do this exactly, right? So that's the beauty of the sort of Gaussian distribution. These integrals are exact. So once you calculate this on a tree, you can then go back and evaluate a posterior distribution for the parameters. So if I were to sort of write in rigorously, it should have been probability of C and V given the observed gene expression values. Now we know that phi, which is fitness flux, is C times V times this tau. So I can just swap V by phi. And the reason we did that is phi is dimensionless. So C goes as 1 over E0 square, and V goes as E0 square if you don't do the rescaling initially, right? But then once you multiply these two, regardless of whether you have done your rescaling properly or not, this quantity will be dimensionless. So it's a robust number to evaluate if you're not very sure about the scale of the traits you're putting in. So that's a sort of a trick we did here. But we were also trying to, so the reason we don't know the scale of the trait is we don't have very good within population sampling of these gene expression values. So we don't have very good handle on the polymorphism. Because of on that uncertainty, we wanted to sort of be sure as what we report as rate of adaptation or fitness flux. So you do your sort of Bayes rule here, and you characterize the posterior distribution for parameters of fitness seascape given the observed gene expression values. The good thing, so as you can see also, I'm not putting the mean here. I put the expression values sampled because you're not going to sample everyone in a population. You're subsampling, so you need an error model to then go from the samples to the true mean. Because this is all probabilistic, all these things can be integrated in it, and you just integrate out all the things you don't know, and so this can be all done in a rigorous way. So this is an example of one of the genes, whatever that is, and posterior distribution of fitness flux and a stabilizing selection. As you can see, oh well, this is lacking a sidebar. So red ones are high. So the rule of the game is basically we can do these curves for every single gene. We can also classify genes by the maximum likelihood values of of adaptation and stabilizing selection. You can go even further and do whatever fancy statistics you want to do. So it's sort of it gives us a way of looking at individual genes separately without having to average over everything.
The question. So here you kind of, before you were doing all genes together, now you're doing each gene individually.
Yeah.
I wonder whether you've done something in the middle. For example, look all the housekeeping genes.
Yeah, we've done that.
You should just
Yeah.
not change much.
Yeah, so we have done all this. I'm not going to show everything.
How if you do the same curve for housekeeping genes?
So you can't do the curve because, well, so we need about couple of hundred genes to make a curve look okay. You can do, for all genes, you can do these measurements, and you can give error bars of your confidence interval, and you can then go and ask what functions they have, and you can classify based on functions, and you can see what genes tend to be more adaptive and what are sort of more under stabilizing selection. So housekeeping genes are among the ones that are strongly stabilizing selection. It would be flat. Yeah, and the things that are under adaptive evolution, so these are the things that we were in particular interested in because there's not much known about them. Well, I mean, people had hypotheses what should be under adaptive evolution, and the way to address this problem has been, okay, I think this is a function that is under adaptive evolution. I go and do maybe some experiments in the lab, see how much I can swap this thing. I will sort of focus on this gene and try to characterize adaptation. So this is already a biased sort of way of doing inference, right? So the way we did this is like, okay, I don't know what these genes are. I just characterize who is adaptive, and then I'll go and see in the literature what I have identified as adaptive, where does it sit? And there's quite a bit of overlap between what we report as adaptive and what people have thought before, but then there are other new things as well.
Are genes with more homologs, paralogs, more adaptive?
We haven't looked at that because that's then tricky to do the alignment there, right?
You don't even do that. You just select subsets of genes.
Yeah.
That are unique in the genome, and those that have rather
Yeah, no, I see your point.
Curious that those that have many
We haven't looked at.
more free to change the expression.
Yeah, that's a good point. We haven't looked at that. So one thing we looked at was basically how level of gene expression determined the amount of adaptation, and that's consistent. So basically, highly expressed genes are on the correct side. They're on the sort of stronger stabilizing selection, right? So these are the ones that have a strong codon usage bias, right? Because you need the good codons to make them highly expressed. And you see a strong correlation between that and the sort of stabilizing selection you infer. So there's quite a bit of sort of biology going in there, but I'm not going to go into all these details. Feel free to ask me. I'm not around much longer, but you can always email or just read the paper. So at the end of the day, you can basically put together adaptive classes, and there's a huge list, and these are sort of the interesting ones that may make sense to some of you. Stress response, growth regulation, mating behavior, response to stimuli. So these are the things that tend to be adaptive. It makes sense. These things change from species to species, from environment to environment, and they better be able to change their gene expression values in response to that. The sort of landscape or seascape that I'm showing here is averaged over many genes. So it's basically what we should see from this curve, and it's a way of characterizing this curve in a more sort of robust statistical way. So you can then compare different hypotheses about gene expression evolution. So this is basically the area that would give you fitness seascape. So you have significant fitness flux of over the 3 point something, significant stabilizing selection of C around 10, and C around 10 means that your curve has gone from 1 here to 1 over this to 10%, right? So it's quite a bit of drop in divergence diversity ratio. So you're very much constrained in evolution, but also very much evolving in response to environmental fluctuation. All right, so these are just the statistics of how these sort of scores of these likelihoods differ. So it's a significant measure, and the significance is not surprising because you just have a lot of genes to put in. So I wouldn't. So at the level of individual genes, their scores are over the 1, so they're not super significant. But once you group things together, you acquire significance.
So what is the fitness flux? What is the fitness flux being 3.5 exactly?
So if you remember, we had basically how likely it is to go forward in time versus backward in time, and that's the e to the 5, right? So it's basically much more likely that you're being driven, right? So fitness flux larger than 1 is basically pretty significant in terms of adaptive evolution. All right, so this was the story that we cooked up as when environment drives an evolution of a population without feedback, right? And that means that the populations don't feedback their environment they live in so that it goes back to them in a way. Now you can sort of go a step further. And this is the thing that a lot of people in the room are interested in, sort of trying to look at co-evolving populations between pathogens and hosts, so things like that. And in this sense, basically what happens is that population A acts like an environment for population B, so it drives the evolution of population B, but then once population B changes, it changes the environment of population A, so you're sort of stuck in this Alice and Red Queen circle, and so they constantly run after each other. They may sort of have a constant distance between each other, but they keep evolving. And the system we started looking at, and we were very interested in it, and we still are, is the interaction between HIV, which is sort of a chronic virus, and this is a picture of HIV. It's an RNA virus. It infects quite a bit of people in the world, and it has all these sort of issues. I'm not going into pathology of HIV, and the adaptive immune system. And so I will argue for you in a couple of minutes why this is an evolutionary process that happens on that happens very rapidly on very short time scales and within the lifetime of organisms, and organisms being us. It's an interesting problem. It has a lot of translational impacts, but also sort of on the mathematical side, it's quite an interesting and challenging problem to look at. So on the HIV side, let me tell you about the evolution of HIV. So far, the time scales we looked at was 4 billion years of evolution. Everyone was evolving. Then Drosophila that we looked was 40 million years of evolution. And this is a tree of HIV over 2.5 years of evolution of the virus within one individual. So a much shorter evolutionary time scale. And so it's the envelope protein of the virus on the surface. It's a region that can easily be targeted by the immune system. So it's always challenged by the immune system. And again, the sort of, as you can see now, there is a component of time. So these are sequences of HIV sampled within individual for 169 weeks, about 3 years. And the time points are sort of marked by color. So from week 4 to week 169. And the distances are, again, mutational distances, and y-axis is meaningless. So basically, yeah, so this is 1% divergence in their genome. If you look at within patient evolution of HIV over 10 years of evolution in one patient, you normally see around 8% to 10% of sequence divergence. So it's a very rapidly evolving virus. This is a number that is equivalent to 8 million years of Drosophila evolution. So a bunch of those species pulled together. And 20 years of global influenza evolution. So if you think that every year we need a new vaccine, basically every person needs a new vaccine for HIV and needs to be updated every couple of months. That's basically what it is. So that's why we don't have a vaccine. So the progression of disease is kind of interesting. So initially, when someone gets infected with HIV, it's an acute phase of infection. So there are flu-type symptoms. They don't know normally that they're infected by HIV. It takes a week, a couple of weeks. And then the virus goes through a chronic phase. And the chronic phase can last up to 10 to 15 years even without therapy. It's a rare event, 15 years without therapy, but it can last for a long time. The viral load here in red is pretty constant during that time, but that's what's going on for the virus. So it's there's quite a bit of evolution during this 10 to 15 years of sort of stable population. Yes?
Maybe you said this, but what is the criterion to say that this HIV evolved into different species now?
So we're not saying it's evolved into different species because the definition of a species is very sort of fake here exactly. Like what do you define it? I mean, so HIV is not even a bacteria, right? So it cannot reproduce on its own. It needs a human host to reproduce, right? One definition of species was sort of criteria of mating, right? Which only applies to basically sexual populations. So that doesn't even apply to bacteria. So let's not talk about speciation. What we can see is basically if you have two on a tree, right? If you have two larger branches sort of splitting at a time and they go independently and evolve independently, that may be evidence of something that might look like a speciation. But so far, at least in this data, we don't see it. In flu, in last year flu, there is some sort of evidence that there is a speciation in flu. So you have two independent branches going sideways, right? So and that's the closest thing that I've seen in sort of viral evolution to speciation. That means that probably next year flu vaccines are not that amazing, or they're very good depending on who is winning. But it was a hard prediction. But here, yeah, so we don't see it unless you sort of want to argue this is something that looks like speciation. So what you can basically just measure is that how much sequence variation you have between two two strains, right? Just by aligning these sequences. All right. So the generation time of the virus, so the reason we see so much diversity. So the virus evolves very fast. The generation time is about 2 days, right? And every time the virus replicates, you get every single point mutations in the genome. So mutation rate is high. And population size is very large, but it's hard to measure it. And that's why the error bars are in the exponent. Because the virus goes into reservoirs in human cells. And if you're in a reservoir, meaning you're hidden, you're not being expressed on the surface, and you're not being detected. So it's very difficult to actually detect a lot of the silent viruses.
How many base pairs?
The envelope is about 1,000 amino acids, so less than 1,000 amino acids. All right. Okay, so that was the sort of bad guys in the story, and then we have talked about. So this was evolution of flu in one individual, right? HIV in one individual. So there's a sort of immune system component to it, and you can talk to Alex and Thierry more about how the immune system works. There's a project going on, and there's sort of students here, Nathaniel, Julia, who work on these problems. So they can tell you much more deeply about how the immune system works, but just in a nutshell, we have genes in our DNA that encode for receptors on our adaptive immune system. An adaptive immune system is unique to vertebrates, although we have CRISPR versions of that in bacteria. And what they do is that you get unique receptors on B and T cells, and they can go and mount a specific response against a huge diversity of pathogens that we see. And the diversity of these receptors come from various mechanisms that are encoded in our sort of genome. So one thing that it is here is sort of this combinatoric alignment of what is known as V and D and J genes. So we have these sort of Legos of V, D, and J genes in our genome in the chromosome 14. So you want to build a receptor at the germline level. What you do is that you take a D and a J, put them together. You put a V, put that as well, and you produce around 2,000 sequences out of this combinatorics. On top of that, you even further diversify this thing by insertion and deletion at the junctional regions of these V, D, and J. And basically, you add some selection against self-reaction to your self proteins because you don't want to attack yourself. And as a result of this sort of insertion, deletion, and selection, you get something around 10 to the 16 or 10 to the 18 sequences. That's much smaller than the number of B or T cells we have in our body. So our repertoire is undersampling what could be produced by the adaptive immune system. And you get the sort of two types of receptors in our body, so B cell receptors and T cell receptors. Both of them are diverse. B cell receptors can detach from the cell surface, and then these are the things that are called antibodies. And they can go around in the blood and attack different sort of entities, lipids, proteins, everything that come in, tag them. And so, but B cells even further diversify, and that's kind of a unique thing. There's an evolutionary process. You start from a B cell receptor that's okay against sort of interacting with a specific pathogen. It can go to a lymph node in our body and get presented by this pathogen, and it goes under cycles of mutation and selection. And during these cycles, it sort of accumulates mutations in a much higher rate, orders of magnitude larger than human genome mutation rate. And if a mutation is good for the cell receptor, it gets selected. So it's a Darwinian evolution that happens in our body. These regions that are formed in lymph nodes last about 2 weeks or so. And as a result of that affinity maturation, you produce the lineages of B cells that are related to each other by this common ancestor, the original receptor you produce, and also all the mutations that have sort of generated during this time. And as a result of this, you end up producing receptors that can be up to 1,000-fold higher in affinity compared to the ancestor you started with. So it's a pretty efficient process. From the data point of view, what you can do is that you can now go in the blood, sample these receptors, and try to cluster them based on their ancestor receptors. So these are all sort of statistical glitches. I'm not going into this. But you see, ensembles of these phylogenetic trees that you can build based on receptors of the immune system in your body. And these are basically, forests of immune sort of species that interact with the diversity of pathogens we interact with. So this is an example we reconstructed from, this is one lineage of B cells, so these are called lineages, that is responsive to we don't know it's responsive, that's collected from HIV patient, right? But it's basically tens of thousands of these trees. It's not only one. For each individual, you can produce that. So the question is, how well our immune system responds to pathogens? Can we, yeah?
So is deselection only against self-reaction, not to specialize to certain?
So no. So the secondary selection in the lymph node, right? It's mainly against the pathogen that is presented in the lymph nodes. So there's a self-selection against, selection against binding to self in the very beginning. In the lymph node, there is a component that keeps cells from becoming self-reactive. Basically, you need T cells to help B cells to express themselves. And if the B cell is very self-reactive, you don't find the T cell to help it. That's more or less how it works. But we can discuss that in detail later. Anyway, so you get, so this 1,000-fold increase is by selection against the pathogen you're encountering. All right, so what I'm going to tell you about this question is trying to go back to what we have built so far, right? Let's try to model the system first. We've recently looked at sort of the repertoire statistics and how numbers actually, from these sort of receptor sequences, how the dynamics of receptor sequences over time in HIV patient work, trying to characterize it with evolutionary models. But for now, let's try to sort of stick to what we've built. So we want to sort of extend our phenotypic evolutionary model to now a co-evolutionary case. And imagine the pink thing is a viral genome or viral envelope, and there are regions on the viral envelope that are called epitopes that basically are targets for B cell receptors or antibodies. So these are the sort of thicker pieces, the colorful. And you can, in principle, target them with different lineages of B cells, right? So that's how it is, right? So they're different in sequence. You need a different receptor to target different ones. So that's basically the picture we're building on. So multiple epitopes, multiple B cell lineages that interact with each of these things. And you can sort of go back to what we built, right? A simple spin model of interaction. And to make life easier, instead of going 0 and 1, we do 1 and plus 1 and minus 1. And you have an antibody and antigen. So antigen is sort of the thing from the pathogenic part, right? And basically, we define a binding affinity that if these two sites are aligned with each other, you increase the affinity, and if they're misaligned, you decrease the affinity. It's just a sort of simple extension of what we already have. So we define energy between, or affinity between, a specific strain of an antibody and a specific strain of the virus. And there's, again, the quench variables epsilon i's that say the contribution of different sizes affinity. Then there are regions in the virus that are conserved. So it's very difficult to flip the spin in these regions. And they're conserved for sort of intrinsic reasons of the reason for the virus. The virus needs to enter the cell, so the receptor that it binds to the cell surface to get itself in should be pretty conserved, because human cell surface doesn't change that much, so you need to you're pretty constrained there, for example. And so for that, what we sort of put in was that there is a conserved region. There's always a spin up in the viral side, and so just a convention, and the antibodies basically can flip to align themselves to that region. So that's the sort of tilde region, which you just by definition put VI is equal to 1. So the reason we put this in was there are antibodies that manage to target these regions, and they're known as broadly neutralizing antibodies, and they're currently the hope for building vaccines against HIV. So if you manage to bind to this region, it's pretty difficult for the virus to escape. That's why we wanted to sort of introduce this, and you can characterize, the potential breadth for an antibody is how much you care about binding to this region as opposed to the other region. Yeah?
Epsilon are plus minus 1?
Epsilons can be any number. They're positive, but you can put them negative. You could put them negative, it doesn't matter, it just tells you that it's random. You can draw it from a given distribution, right? So at the end of the day, you do some sort of anneal diverging over statistics of your disorder in the system, which are your epsilons. They're sort of quench They're quench variables, right? And so that's basically what you do. Being positive or negative basically tells you if you favor parallel or anti-parallel. So it's just a change of convention. It doesn't matter. All right. Right. So if you think of lineages binding to specific regions, right? Yeah, so like L would be the whole length of the genome. A lot of epsilons are 0, right? So and then maybe for this lineage, only the black region is non-zero. So that's basically what you care about. Stability for antibody. So if you have an antibody with positive or non-zero epsilon tildes, means that that antibody lineage has the potential to interact with the conserved region. Right, so it matters, what are your potential. Still, if you have a non-zero epsilon tilde but pretty crappy AI alphas, you're a bad antibody, right? So you still need to align yourself, but you have a potential to see the region, right? So the things that you can change is, yeah. So AI, you need to evolve your AIs, but if you don't see the region, you can't evolve them accordingly. The A's, yeah. The green term is binding to a region of the virus that the virus doesn't change. So it's always a spin up. So it's a conserved region of the virus. So the model is sort of both sides can evolve, A and V can evolve, but there's a region the virus cannot evolve in very well. So a conserved region, so that's the tilde region, the green. And or 1, yeah. So they're always a sort of.
Like a random field?
Okay, so the epsilons are the random field quench variables, right? So, but then by evolution in the viral antibody sides, right? So it goes in different direction. Antibodies, the A's, align themselves to whatever the V's are. The V's try to misalign themselves to whatever the A's are. So it's sort of, there's a frustration in the system. So that's what sort of the left-hand side will end up being doing, right?
I don't think epsilon is an external field, because epsilon is in both terms.
No, epsilon's still the.
It's just the weight of this speed.
No, but it's fixed, right? And it's fixed externally, so you can't. No, it's different. It's epsilon, epsilon tilde. So there's no pairwise. So there's no pairwise interaction between sites, right? So between A and V. So it's basically still linear for each of these, right? So if you fix the virus, whatever V's are, then that sets what your field is exactly. And then you try to align your A's to whatever that V is. The only difference is basically the dynamics in this region will be quite different from the other region, because here you have frustration and here you don't. You can sort of, you have a single solution on the other side. Okay. So you can look at the distribution of, again, binding affinity. So for each of these antibodies and viruses, you can sort of look at the distribution of its binding affinity against all the circulating viruses that are in there, right? So now instead of one number for each antibody, you have distributions for each antibody, and the same way you have distributions for each virus. Now you can sort of pick the rules of the game as you wish. You can say, I care about average affinity, I care about the best affinity in the system, and you can solve various versions of the problem. So the mean affinity is just, you weigh these viruses by their frequencies, and then you calculate the mean. The best affinity, you can use extreme value statistics and sort of look at the tail of the distribution and pick up what's in the tail. Then in that case, you care about how many viruses you interact with and what's the variance of this virus in a sort of so the number of interactions you deal with comes logarithmic, and the variance comes as square root of here, right? So for the time being, let's just stick to the simplest case. I care about the mean affinity. It's not the best example, right? But you can solve both. It's just to move forward. And you can define, once you characterize the phenotype you care about the antibody, then you can look at the distribution of that phenotype now across all the antibodies. And that's the thing that sort of your quantitative trait or molecular phenotype evolution will act on. Then you can do the exact same thing for the viral side. So you have two distribution of phenotypes for each of these strains, each of these populations, and the idea is that antibodies want to increase their affinity and viruses want to decrease their affinity. And you have a joint stochastic dynamics between the two in a form that it doesn't split like the thing we wrote here for environment driving the virus, because you have the feedback between the two. So you sort of build up these propagators. And again, you put in all the ingredients of evolution. I'm not going to derive anything for this part. It's really very similar to what we have done before. So let's say we have mutations, we have genetic drift that take you from this, and these happen independently in the two populations. They come from random mutations, and they come from sampling. And then you have selection that is what is known as antagonistic selection. So increase for affinity in the antibodies. And as you remember, the increase in the affinity is proportional to the diversity. So the speed of change in affinity should be proportional to diversity in each population. So this one, in this case, would sort of move faster than the viral population because it has a larger diversity. And also depends on the selection coefficients. And sort of, in this case, the simple sort of linear selection, and you can do a quadratic one as well, this guy will increase the affinity and the antigens will decrease the affinity. But sort of once you're sort of doing the evolution, you're just flipping the spins, right? This is just telling you how the effective phenotype you produce is impacting your fitness, but the sort of movements you're allowed to do is the spin flips. So you can put together exact same theory and try to solve it, and you write sort of the Langevin type equations for changes of the mean phenotype, the variance of phenotype in each population, and you sort of have the whole series of equations again, do moment closure and all these things. And you can also, going back to what these conserved regions and variable region was. So if you look at the conserved region, so this is a general form, but if you look at the conserved region, the difference between conserved and variable is that you don't have the viral evolution in the conserved region. So you're sort of not changing the affinity of the conserved region on the viral side. So that's basically the only difference. There's no co-evolution there. It's a transient dynamics that takes you there. what makes it non-trivial is that the conserved and non-conserved regions are linked to each other, and you have many lineages in the population that at the same time want to do something good for themselves, right? So if all of a sudden you have an antibody that has a potential to bind the conserved region, but you already have solutions against the virus with other antibodies, the broadly neutralizing one might not succeed. So, and that comes from what's around in the population. All right, so let's skip that. We have red queen dynamics. All right. We can go back and try to, again, characterize the sort of fitness flux notion in the system. The total rate of adaptation can be defined in a very similar way. The difference here from what we had before is that we have a common molecular phenotype, which is the affinity, and it impacts the viral and antibody fitness in opposite direction. So antibodies want to increase the affinity and viruses want to decrease the affinity. So if you're at this point and antibodies want to evolve, they go up the fitness peak, fitness hill here, and so you gain a fitness by the blue arrow, and you do this over time. The sum of these blue arrows will give you the fitness flux, or as amount of sort of entropy production in that system. Then, but there's something else also happening, is that if your antibodies adapt and they go up, the fitness of the virus just drops without viruses doing anything, right? So that was we're sort of fixing the virus and letting antibody to evolve. The fitness of the virus drops because the phenotype is now increasing. And the drop in the fitness of the virus is just a transfer of the fitness from antibodies to viruses. And that's what we call now transfer flux. That's a measure of interactions between antibodies and viruses. And if this guy is a heat dissipation, as we argued before, transfer flux basically plays the role of work in the system. So it's the work that antibodies do on the virus without viruses actually doing anything. And in the notion of population genetics, work is meaningless because what we so far discussed before, right, is meaningless because we don't have conservation of energy or anything that looks like conservation of energy. But here, because we have a coupled system and we are detaching pieces of system from each other, it's actually meaningful because work is meaningful in sub parts of your system, but not in the whole system together. All right. So you can sort of characterize the amount of adaptation or escape by phenotypic diversity. Because rate of adaptation and rate of sort of interaction depends on the diversity of each side, as we had before. All right. So how can we quantify this? Before we had this sort of divergence diversity curve that tells us what fitness flux should be. So an interesting quantity to look at to characterize both fitness and transfer flux is by looking at time-shifted fitness measurements. So if I take an antibody or a virus at a given time point and I measure its fitness against, let's say, I'm taking a virus today and I take its fitness against antibodies that appeared in the past and antibodies that would appear in the future, right? So you would expect that if viruses have escaped something in the past, they're pretty fit against those antibodies, right? So that's basically what they are trained against. And if antibodies are now going to neutralize this virus, so if you sample some antibody in the future, these are the ones that are pretty good against the virus, so the viral fitness should be pretty low against future antibodies, right? So if you have a balanced coevolution, as you expect, you get these sort of S-shaped curves for time-delayed fitness of the virus. You can characterize the shape of the curve, and what you see is that the slope, in the stationary state, the slope at time 0 is the same because you move as much as the other guy gives you space. But in principle, what you end up having is that the slope of the curve towards the past gives you fitness flux, because that's the amount you adapt to the stimuli in the past. And the slope of the curve towards future gives you the transfer flux, because that's what the other population would impose on you by evolving. Is this clear? All right. So if you have a sort of non-co-evolving system, you basically don't get a drop in the future, right? So there's nothing that cares about, right? So you don't have a sort of you don't have a population that with feedback cares about you. You only sort of adapt to what you have seen in the past. You don't necessarily deal with the future, and basically you get a peak in this direction with the fitness flux that would be characterized similarly with a divergence diversity ratio. All right. So you can go and now look at sort of data in HIV patients, right? So these are sort of 2 HIV patients that you measure the viral fitness against the plasma of these patients across different time points. And this is basically what we have managed to characterize. So in one patient, you basically see there's a somewhat evidence for balanced evolution of the B-cell repertoire with the virus. And in the other patient, we don't see much response. And these are actually sort of consistent with then the state of therapy that they would go to, because this guy was not really responding to, his immune system was not that great at the time. But so far everything we have basically put together is the way of building intuition about this co-evolving system and trying to understand things in this setting. All the models we do is for hoping to build sort of a predictive model for dynamics of the repertoire, and more importantly, trying to predict the fate of a broadly neutralizing antibody that might emerge in a population given the state of the sort of repertoire at a given time point. That's important because if you want to sort of get a vaccine against HIV, it's not enough to get any antibody. You need a specific type of lineage that targets the conserved region. And the question is, how likely it is to do that? And the reason that it's complicated is we have quite a bit of competition among lineages in our repertoire. So I mentioned that there are germinal centers, so this GC, that basically are formed during affinity maturation. And the pictures that I'm showing you here are multiphoton microscopy of using rainbow techniques in Gabriel Victoria's lab to stain different B cell, naive B cells that undergo affinity maturation in a lymph node. And you can go under microscope and look at sort of these colorful B cells and look at the dynamics at different time points of sort of the pictures that you see under microscope. And so this is a lymph node that you see. And these are sort of colonies of these BCRs that initially are pretty diverse because you just express different colors in there. So that's day 3 after introducing some artificial antigen in a mouse. And after sort of day 15, what happens is that you have the clonal antibodies that expand in different parts of this lymph node, right? So these are all cells that have originated from a red sort of marked cell in a B cell. So that's basically, you see quite a bit of sort of convergent evolution in a way, or exclusion by competition in each of these sets of germinal centers in the lymph node. But then there are groups that are still quite diverse, and they haven't figured out who is going to win, right? So you said sort of in the third guy, so in this group, you see still quite a bit of diversity maintaining that. So this is the evidence that you have short-term competition among these BCRs within sort of lineages that form the germinal centers. Now, you can do also reinfection. You go back and a couple of weeks later, the mouse is happy. You re-inject the same virus or same antigen again. And then you can see basically germinal centers forming. Now, from this process, you build up memory in your immune system. That's why you vaccinate people, right? And so now you have a long-term competition between the existing memory cells that you have here and also the new responses that you form. So now you go back and you sort of look at the other leg of the mouse, that the new infection is happening in there, and so the memory goes from one side to another side, but you also still see sort of competition between now the existing memory cells and the new groups of cells that happen. So you have short and long-term competition among cells. It's basically like asexual evolution with clonal interference and clonal competition among different lineages. And on top of that, within each lineage, you have still quite a bit of competition. And what you can do is you can try to characterize the sort of fixation probability or the probability that a specific lineage that starts up from a certain frequency takes over or come to a high frequency in a population. And this is basically done by characterizing, let's say you start off with a frequency row of a specific clonal lineage. Its dynamics is proportional to the fitness of that clonal lineage with respect to the rest of the population. It goes back to what we have already. There's no mutation from one lineage to another because you can't change what you care about, right? So these epsilons, these are quenched. The other component that plays a role is the sort of stochasticity, that is small number fluctuations. You put this together, these are subject to dynamics, right? So now all the fitnesses are time dependent because the virus changes, the state of the population changes, so you have to do quite a bit of sort of calculation here, but what you're caring about is basically asymptotic behavior of this frequency in expectation value across ensembles. And there's sort of ways of dealing with this, and probably Michael Desai might tell you something about it, so you need to do Laplace transform of these equations, but at least that's one way of calculating this. But the solution that you end up having is basically you can characterize fixation probability of a clonal lineage compared to what you expect to happen in neutrality to be equal to the relative growth rate at the time of introduction, the relative fitness flux at the time of introduction of the lineage compared to the rest. So the first one is like, who is growing faster? The second one is who has the potential to adapt faster, right? My fitness flux is larger or smaller than the other guys. And the last one is the transfer flux. So who is more vulnerable to the viruses? So this is basically all estimated at time 0, and this is asymptotic behavior.
You're winding down.
All right, this is the last figure. And once you put this down, you can basically characterize fixation probability for broadly neutralizing antibodies versus the sort of normal antibodies as a function of viral diversity. And the viral diversity comes in because you have this transfer flux component. And what you see is that in the sort of low viral diversity regime, these two are more or less the same. But then as viral diversity increases, broadly neutralizing antibodies tend to be more successful in establishing themselves in a population. And the reason for that being is that these are basically generalist solutions to a very diverse viral population. And if you want to tackle that with specific antibodies, you need to basically produce specific receptors for every sort of clonal side of the viruses, whereas for broadly neutralizing antibodies, you have one solution for all. So the only difficulty is that it's just fewer of them and it's harder to make these things. But once you have that, given the conditions are proper, so we have enough viral diversity, you have a good chance of fixing them. I think I'll skip this. And with this sort of, to recap the second part, so we basically we extended this phenotypic evolutionary picture to now a co-evolutionary picture. And the time-dependent observables in this case would be the time-shifted measurements of fitness. We have fitness and transfer flux that characterize heat dissipation or entropy production and work in your system, rate of adaptation and interaction. You have BNAbs, broadly neutralizing antibodies, that can be induced by controlling viral population diversity. And the thing that I didn't show is broadly neutralizing antibodies tend to circulate longer in a population without going extinct and by reaching higher affinities. And that comes from the fact that it's harder to go extinct if you are not subject to escape or vulnerability from the viral site. And with this, I think, okay, so we're not going into those things. Close the lecture, and I hope you learned something about this sort of evolutionary processes. I don't know.